Hey guys, it's me. Mario apparently. No, okay, it's Naomi, and I'm gonna stick with that joke. Um, so this is the third content block of your final biology review, and in this one, I kind of just want to go through general biology exam techniques, so things that will help you get those marks you deserve. So we'll be talking about multiple choice questions, short answer questions, how to draw diagrams, how to write up pracs, and how to study for biology because it's kind of very different from studying for things like English and maths. So let's get started. So I've had a lot of people ask me whether they should do multiple choice questions first or last, and I generally always say first, and that's for a couple of reasons. Number one, kickstarts your biology brain, gets you thinking in the right mindset, particularly if you've had an exam the day before. Um, Number two, I think it's important to get them done first so that you have an informed understanding of how much time you have left. If you're trying to write the answer to an 8-mark short answer question, but you have still got so many marks of multiple choice questions to go, you don't really have an informed understanding of how much time you actually have to spend on that short answer question because you don't know how long the MCQs are actually going to take you. There could be some tricky ones in there. So I think it's important to get them out of the way so that you can have an accurate estimate of how you're going time-wise. I also think that often MCQs contain examples or definitions or content that can help you answer the short answer questions. So it's a good idea to get them done first and have all that content in front of you, which may actually help you with questions later on. If you really don't know the answer, you can generally always rule out one or two, leaving you with an increased chance of guessing the right answer if that's what you've got to do in that case. If you're really not sure of something, though, I would say mark it, come back to it later, and have another go and see if maybe you read it incorrectly or your completion of the short answer questions has given you more insight into what the question might be asking you. Speaking of what the question might be asking you, I think a good rule of thumb is to ask yourself what the Board of Studies is actually testing you with the multiple choice question, especially if you really don't know. So let's look at a question so I can show you what I mean by that. So nitrogenous waste is its highest concentration in the renal vein, the renal artery, the collecting duct, or the cortex. So this is really testing your understanding of the function of the kidney and the structure of the kidney. So if you think about the function of the kidney, one of which is removing nitrogenous waste, we know that and combine that with your knowledge of the structure of the kidney, which we know is composed of nephrons, along which is the convoluted tubules, the proximal and distal, and the collecting duct, which leads to the ureter, the bladder, for the nitrogenous waste and urine to be urinated out. We can pretty much conclude that the answer has to be C, because the collecting duct is furthest along the nephron of the kidney, where the most urea will have built up by secretion into the nephron. So now let's talk about SAQs. So the first thing you have to do in any short answer question is look at how much it's actually worth. I always tell students who write, paragraphs and paragraphs, way too much for 2-mark questions, that you can't get more than 2 out of 2 for a 2-mark question. The most you'll get is 100%. So you need to look at what the question is worth and write the amount that will get you all the marks. So for a 1 to 3 mark question, what you're generally going to be expected to do is clearly define all the key terms in a 1-sentence definition and clearly answer the question. Some 3-mark questions will ask you for an example, in which case provide an example and show why it's relevant, and draw a diagram if they ask you to or if they leave you space. Don't do more than that. You cannot get more than 3 marks in a 3-mark question. Do not waste your time. In a 4+ mark question is where things start getting tricky and the lines start getting blurred about where marks are earned. What you will always need to do is define the key terms, provide a clear and comprehensive answer to the question using the words of the question, provide examples. Even if you're not prompted and you know an example, provide one. And if there's space for a diagram, or if you can draw a diagram that's relevant, draw it. That is where the marks come from. So now let's talk about those really high mark questions and a bit more in depth about how to answer them to get all those marks. So the first rule of thumb is always address the verb. Don't don't discuss something if they ask you to justify it. And if they ask you to compare and contrast something, don't just define the terms. You always have to look at the verb in question and see what they're asking you to actually do. First rule of thumb, like we always said, define everything. There's always marks in there for definition. You will lose out. Even if you think, oh, they already know what evolution is. Oh, it's not a key part of the question. Just define it. Have a one-sentence definition and just do it. So, people often ask me whether they're allowed to use tables. Yes, you are absolutely allowed to use tables. In a compare and contrast question, or a contrast question, or anything that's about the differences between things, tables are a fantastic choice. They make your wording, your understanding, the ideas you're trying to express really clear, and it essentially gives the marker a place where they can tick, which is really what they're looking for. Tables are absolutely fine. You can even use headings and underline key terms. So if you're trying to get a point across, if you're trying to, make 3 points in an 8-mark question, you should absolutely underline the keywords, or even kind of write a number to show that you're providing 2 examples. Number them 1 and 2. Show the marker that you've completed all parts of the question, and headings and underlining is absolutely appropriate for that. Draw diagrams. So sometimes the tests will leave you a sneaky bit of blank space below marks, below your lines, to kind of give you a clue that you're supposed to draw a diagram for that question. And definitely draw a diagram in those ones. But even questions that you have a diagram for, that you think a diagram would better show your understanding, draw one as well. Definitely answer the question in words, but feel free to draw a diagram. There are marks for those. Essentially, like I've been saying, do anything that proves to the marker that you have answered all parts of the question. Use keywords, underline the main points that you want them to get, use tables, anything. Just show them that you've answered the question, that you understand what they want you to say. Let's talk about diagrams now. So they should always be in pencil, should always be labelled. I like to label the variables, so I like to label the independent and the dependent variable and things that are controlled, so that they know that I know about experimental design. No shading. The amount of students I've seen who've shaded in diagrams, that's not what we're looking for. We definitely don't want shading. So just draw an outline of what you're doing with a pencil and label it with a ruler. Like I said, so the main point here, i.e. the variables, you can circle and underline them to show that you understand the purpose of the experiment, that you're not just setting up 2 beakers for, Lord knows why, but you actually understand the point. So I'm sure you can tell me by this point which diagram is good and which one is not. We'll quickly go through them. Here we've got some shading, clearly no ruler used. They haven't labelled anything. It's in pen, just a disaster basically. That's not very many marks. Here we've got a diagram drawn in pencil with a ruler, labelled clearly. We've shown it's a swan-necked flask. We've underlined here the key variable, the key outcome of the prac, which is this one has no pathogens in it. We've labelled all the key parts. So this is a full marks diagram. Now let's talk a bit about experimental design. So pretty much any experiment they give you, unless they already give you part of it, you should be writing an aim, a hypothesis, your materials used, your variables, your risk assessment, your method, and if they specifically ask you to, you should write about the results and the discussion. You can't really write, and probably the conclusion as well. So if it's an experiment that you haven't done in class, there's no way they could ask you to know the results of the experiment. So it's highly unlikely that it'd ask you to write results or a discussion or a conclusion. But you will always be expected to write these up. So you should know them well for all the pracs that you've done in class that are in the syllabus. So let's check our understanding of experimental design and all the variables. So let's read this one from 2015. It's a 3-mark question. Students wanted to test whether the height of plants decreased with increased elevation. They planted 10 plants at varying elevations at the same mountain range and watered them the same amount. The graph below shows the average height of plants at their locations. Evaluate the experiment's validity. So, being able to answer this question properly requires you to understand the meaning of the word validity, which is essentially, are we testing what we say is being tested? Are we testing whether the height of plants decreases with increased elevation? So there's a few things to think about when we're evaluating validity. So we've already discussed what validity is. Let's talk about what's good about it here. So we've got 10 plants at varying elevations on the same mountain range and watered them the same amount. So that's good about validity because we're controlling some variables by making sure it's the same mountain and the same water. There's one really obvious thing that's not good about validity here, but you've got to look carefully to see it. And that is, right over here in the top left-hand corner, sneaky, the prevailing wind. Even though these plants are planted on the same mountain range, they are exposed to different levels of prevailing wind. The wind is not being controlled when it should be. And this impacts the validity of the experiment. Because we're not just testing changes in elevation, we're also testing the effect of changes in wind. So we can see that the effect of wind may impede validity. So you can quickly talk about the good things and the bad things. I would say overall that the experiment is not valid. Great. So let's just quickly go through the definitions of things you need to know. This is one of those slides that you should probably just screenshot and memorize. So validity is about how well the method of the experiment is designed to test the given aim. So that includes proper variables, and your other variables have to be controlled. Reliability is how consistently the results of the experiment can be reproduced. So if you give your exact experiment to someone in Sweden, will they be able to copy what you've done and get the same result? What that's about is repeating the experiment a whole bunch of times to make sure you get the same outcome, excluding any outliers that occurred due to errors or just random variation, and finding averages of results. Accuracy is how close the results of the experiment are to the results you expect if performed under perfect conditions with perfect tools. So that's about having really good measuring equipment and avoiding human error. So reading all the measurements properly. Great, so now let's talk a bit about how to study for biology. So I'm a huge advocate of past papers. I genuinely think that the best thing you can do for yourself is do as many past HSC questions as possible. You guys are in luck because you're one of the last years to be doing this current syllabus. So you have one of the biggest stocks of practice papers that any biology year has had. So pretty much do all of them. They're all available online, and I know that ATAR Notes also sells question books that you can buy online. Me and a friend, Maddie, wrote those. But essentially, there's no reason why you shouldn't have access to practice papers, and there's no reason why you shouldn't be doing as many as you can. For me, I did one paper a day in the weeks up to the Bio HSC. So, essentially, try to do as many as possible. And as you get better and better, you'll find that they're not taking you the full 3 hours. I think by the end, if I were to rush through a paper, answer every question, but do it quickly and not check my work, it was taking about 2 hours. So I would say that there's not really an excuse for not doing as many practice papers as you can. It's the best tool for you. Again, I think you should write out the answers to SAQs every time. Even, if you've written out the definition for evolution 5 times, the more times you write it, the more fluid it will become, and the more able you will be to write it in an exam situation, which is super high stress, and the more capable you will be at using that definition to answer a question. So the more you write it, the better you'll understand it, and you'll better understand your knowledge, the utility of your knowledge in a biology exam. I would also say do all the multiple choice questions every time. Even if you think you know the answers or you think that they're easy and you always get, 18 out of 20 or whatever, do them all every time. You may get some wrong. You may make a silly mistake, and you should always mark them to make sure you know where you went wrong and go and read up about why you got it wrong and work out what the correct answer should have been and why. Cool. So memorizing for biology is hard. There's a whole lot of content, and it's hard to get it all in your head, particularly when you've got, another 5-6 subjects to think about at the same time. So the technique I used was palm cards. I condensed the notes that I'd written into really comprehensive palm cards, which I used color coding in. I highlighted things, I underlined things, and you may have noticed these 3 content blocks I've been using red for science and blue for scientists. Red for technology, and blue for scientists. I did the same thing with my palm cards. Everything was color-coded, and that really did help to jog my memory. You can sort through and organize the content in your own head and your own way. And importantly, I love to put things on low or high rotation. What I mean by that is that I would flag palm cards that I was having the most difficulty with that were also really high yield content. I'd flag them with a red post-it note, so I knew that I would have to come back and read them more often than others to be able to understand the content and get it to stick in my head. Ones that I'd flagged with a green post-it note, I was pretty confident with, and I knew that I didn't need to read as much and study as much. And that was how I was able to prioritize my learning. Again, color coding, like I was talking about, they're really portable. So you can just, have them in your pocket on the bus, take them out, have a read in, a spare 5 minutes or during school assembly. Or you could listen during school assembly. Read your palm cards. Great. And also the fact that a summary, a one-sentence summary of an idea that you do understand is much less intimidating than trying to memorize a page of information about something. Great, exactly. So you see all those, horrifically scary pictures of people's study notes where they've, written in this insanely neat writing and everything's all color-coded and beautiful and perfect. It doesn't need to be pretty. You just need to be able to learn it. So I think I've got for you here. Okay, here we go. So active reading is something I really promote, which is essentially, at this point, I would say that it's probably too late to start writing your own notes. It wouldn't be an efficient use of your time, particularly when you've got so many other subjects to choose from. So find some notes somewhere if you don't already have a set of your own, and read through them actively. And what I mean by that is you should have a pen and a highlighter right on hand. You should be annotating them. You should be highlighting key parts. You should be flagging things that you need to come back to. You should make the notes your own. And I think that will definitely help the information get into your head better than passively sitting there just reading it, highlighting every single word, which I think we've all done at some point. Cool, so here's an example of the palm cards I was making. So you can see over here, I've got all the people, what they did and how they did it. And you can see that I'm not using too many words. I'm not going into insane levels of detail. I'm using just enough to jog my memory such that I could answer a short answer question about it with enough detail. I've also got a description of the prac, and it's all labelled and what I used. The benefits of models, which you'll see is in the syllabus as well. Table here that I could very easily replicate in an exam, and this to help me understand what exactly is going on in meiosis. Great. Now let's quickly talk about some general HSC tips, and then I'll let you be on your way. So I think the most important thing is it's really easy to get anxious about the amount of exams and the amount of content. And it's easy to stay calm, to say stay calm in hindsight. But I think the most important thing is to understand that, if you prioritize your time and you study effectively, you will get the content in your head, particularly for biology. It is very, very memorizable and a lot of content overlaps. So you will be able to do this. I think you should establish a study routine. So work out for yourself exactly how many hours a day you want to study, when you want to start studying, when you want to take breaks, and when you want to stop. Otherwise, there's nothing to stop you from sleeping in until 1 and then studying until 4 in the morning, which isn't healthy if you want to be awake during your exam. And there's also nothing to stop you from being anxious during lunch because you feel like you're wasting time eating lunch and not studying, which also isn't healthy. I often have problems focusing at home, and so what I do is I go and I change my scenery. So I often go to the library or to a park or to a cafe and study there, and I find it, particularly in the library with people studying around me, it's easier to focus. I think if you're struggling either with content or with just coping with the stresses of the HSC, you should definitely ask your friends or your teachers for help, and they'll be able to provide you with some level of support. You should also send in practice essays. So particularly for subjects like English and history, you need to be doing as many practice essays as you possibly can. So send those in either to your teacher or, as you know, hopefully to atarnotes.com where you can get your essays marked for free. So just to give you an idea of my study strategy during StuVac, the first thing I do in the morning is get the most difficult thing out of the way. And for me, that was getting a whole English or a whole history paper done. I'd always find it really hard to actually sit down and just grind out an essay. So I made sure I'd do that first before I got on with my day. So that would be 2 hours of full English paper or 3 hours of full history paper. I'd just I'd wake up, I'd make myself a coffee, I'd sit there and I'd do it. Afterwards, I'd reflect on what I'd written, I'd highlight any good sentences I'd used, and I'd work out how to improve my argument for next time or where I went wrong. Then I'd have breakfast, and I'd give myself really, probably an hour and a half to really relax and just chill out, That would be when I'd check my Facebook, check out the news for the day, go for a walk if I wanted to. Then I'd go to my study space. So sometimes that'd be at home, sometimes that'd be at the library. And there what I'd do is I'd revise my notes. This is the time that I'd actually memorize my biology notes or my English quotes. I'd get stuff into my head. In the evening, I'd do a bio or a chem paper, which would take 3 hours. It got less towards the end. They started to take 2.5 hours, 2 hours, and I'd always mark them. You have to mark your paper so you know where you're going wrong. Then, in the evening, I'd have a chill, relax a bit more, and then right before bed, I'd go through my palm cards or I'd go through my notes that I'd annotated and I'd reread the most high-yield content to make sure that it went into my head. And this was an effective strategy for me. Anyway, I guess that brings us to the end. Good luck, guys. I really think you will smash it and you'll be able to get there. So yeah, all the best for your exams.